All right, so we'll pick back up here with part two. We'll start here by talking about the trachea. So the trachea is going to be located just inferior to our larynx, and it again is going to be only involved with the respiratory system. So it's only going to get in air normally. Now, there is a strong emphasis in a clinical setting to prevent something called aspiration. And now aspiration is when you take ingested material, so food or liquids or something like that, which normally travels down the esophagus, and aspiration is when it bypasses the epiglottis and actually enters into the trachea. So this is a very strong clinical correlate, and it's something you want to try to prevent at all costs. Our trachea is often referred to as our windpipe. It's anywhere from 4 to 5 inches in length, about an inch in diameter, and it contains all of those different tracheal rings. Which, do those make a complete circle? Hopefully you're saying that they do not. So they're C-shaped, so they're open. They're open in the back. Now we have different layers of our trachea. So we have a mucosa, a submucosa, and then kind of an adventitia, which is going to be our outermost layer. So our mucosa is going to consist of epithelium, the ciliated pseudostratified epithelium that we mentioned in part one. And the cilia specifically will function as a mucociliary escalator. And so if you think about what this word means, muco is referring to mucus, ciliary is referring to cilia, and then escalator is involving, bringing something up like an escalator does. And so this cilia is going to beat rhythmically and slowly begin to push mucus that has trapped dust particles and foreign debris and possibly bacteria or something like that that's trapped in this thick viscous mucus. This mucociliary escalator will function to move it upward out of the trachea, and it will ultimately enter into one of two places. It'll enter into our mouth, and then we can do one of two things. We can either choose to swallow it and it goes down our esophagus into our stomach, or you can choose to get rid of it and spit it out. And so either way, you're getting rid of this mucus with its trapped debris. Just beneath that, so just beneath the mucosa, you have the submucosa, which is named just because it's located just beneath the mucosa. It contains areolar or loose connective tissue. More external to that, you have your adventitia, which again is still loose connective tissue that kind of helps anchor the trachea to surrounding structures within the body. And then those tracheal rings that I mentioned, you have anywhere from 15, 16 to 20. They're all C-shaped that open to the back. And they are connected, so they open to the back, so there has to be something to make a connection between them. And it's connective tissue in a muscle called the trachealis muscle. So this muscle is named according to where it's located, so it's associated with the trachea, so we'll just call it the trachealis muscle. Now, the piece of cartilage, the tracheal cartilage ring, that is located at the junction of the trachea dividing into our primary bronchi, this is going to be called the carina. And so it has a very distinct appearance that allows you to tell it apart from other ones. So here we have so you have your larynx further up here. You have your trachea coming down with all of your tracheal rings. And again, you can see on this right side, they are not connected posteriorly. They're connected via this trachealis muscle and different connective tissue. And then here down at the end, this is your carina right here. So it has a different shape than your other tracheal rings, and you can kind of see it on this right image as well. And then this is the point where our trachea will divide into our primary main stem bronchi. So your right and your left over here. So here's a micrograph of our ciliated pseudostratified columnar epithelium. So again, we see our nuclei that are staggered. All the cells still touch the basement membrane down here. You see a mucus-producing goblet cell in the middle, and you can see your cilia on top. So as this goblet cell secretes mucus, it secretes it onto the surface. It is contained within, the cilia are contained within it. That mucus will trap any foreign debris, dust particles, anything like that. And then those cilia will beat rhythmically and move it up toward the pharynx. So as we branch from the trachea and we begin to go down, we get into a couple different structures. So we get into our main stem bronchi. So in the clinical setting, oftentimes they're referred to as the main stem bronchi. You can just call them a primary bronchus if you'd like. And these are going to contain or be involved in what we'll call conducting zone structures. So if you think about what conducting means, you're just moving things along from point A to point B. And that's exactly what we have all the way from our main stem bronchi, so our primary bronchus, all the way down to a level we'll call our terminal bronchioles. Everything in between those, including those, are going to be conducting zone structures. And so all that means is that air is simply being conducted through this series of branching structures and no gas exchange is happening. So we enter into our mouth or our nose, through our pharynx, our larynx, our trachea, into our primary bronchus, secondary, tertiary, bronchioles, and then terminal bronchioles. During all this time, no gas exchange is happening. It is just simply moving down the line. The air is simply moving from the primary to the secondary and so on and so forth. Sometimes your secondary are called lobar bronchi and your tertiary are called segmental bronchi. And these, each lobar bronchus or secondary will supply a lobe of a lung. So on the right there are 3 lobes and on the left there are 2. And then each tertiary or segmental bronchi will provide air to a bronchopulmonary segment. And you have roughly 10 in each lung, so roughly 20 total. Sometimes in the left lung you can have 2 fused together, so you end up with 1 fewer. But in general, you think of there being 20. Now, here we can see our trachea. We have a primary, a secondary, tertiary, and then we get down into bronchioles and terminal bronchioles down here. All of this is all conducting zone structures. So is any gas exchange happening? No. No gas exchange is happening until we get down to our respiratory bronchial level and then into our ultimately our alveoli. So that's where we'll start. We have our respiratory zone structure. So here we do have gas exchange beginning to happen. Okay, we start with respiratory bronchioles. We go to something called an alveolar duct, and so an alveolar duct is just leading into an alveolar sac. And then what contains and what makes up an alveolar sac is all of the individual alveoli, or an alveolus is singular. Now, you can picture and imagine your alveolar sac as a cluster of grapes. So picture a cluster of grapes. That whole cluster, that is the alveolar sac. And then if you pick just an individual grape, one grape is an alveolus. Okay, so hopefully that makes sense. And then the stems of the cluster of grapes, that can represent your respiratory bronchioles and your alveolar duct leading into it. Okay. Now, down at the alveolar level, we have our alveolus and we have a capillary that run right next to each other. The endothelial cell of the capillary and a cell we'll call a type 1 pneumocyte of the alveolus, those two together make up the respiratory membrane. And now the respiratory membrane is extremely, extremely thin. It's on the order of 0.05 micrometers thick. And it's going to exist way down here at the level of our alveolus and the capillary that runs around the alveolus. So here you can see this is your cluster of grapes, if you will, the alveolar sac, and then one individual grape that is going to be a single alveolus. You have respiratory bronchioles running down from your terminal bronchiole. From there you have a alveolar duct running into an individual alveolar sac, and then you'll ultimately get to your alveoli. So I mentioned type 1 pneumocytes. So there's type 1 and there's type 2. Type 1 are going to be simple squamous. So if you think about what simple squamous means, simple is referring to the fact that there is one layer, just a single layer of cells. And then squamous is a flat-shaped cell. So if you think about how this relates to gas exchange, it's pretty intuitive and it makes good sense. A very flat cell that's only one layer is not very thick, and this is perfect for having gases cross these cells, so from the blood into the alveoli or from the alveoli into the blood. So our type 1 pneumocyte will help to comprise the wall of our alveoli and be involved with gas exchange. Type 2 pneumocytes, these are going to be still simple, so a single cell, but they're going to be cube shaped, so they're going to be cuboidal cell. And what these do, they're not necessarily involved in gas exchange directly, but they secrete something called a surfactant. And surfactant is a phospholipid-based compound that coats the inner lining of our alveoli, and it reduces surface tension. So normally there's a little bit of water coating the alveoli, and the water on opposite sides can attract each other, and it can cause the alveoli to kind of fall in on itself, and so this increases surface tension. When you coat it with this surfactant that's produced by these type 2 pneumocytes, it decreases that surface tension so that the lungs are able to expand with a lot more ease. Now, this comes into play when you talk about premature infants, infants who are born prematurely. Their lungs have not developed to the point of having type 2 pneumocytes present. So if they don't have type 2 pneumocytes present, what will they not have? Well, they won't have any surfactant. So if they don't have the surfactant, well, now their surface tension has increased within their lungs. And they can have something called IRDS, or infant respiratory distress syndrome. So you can give them, premature infants will be given an injection of a corticosteroid, and this will increase surfactant production that will aid in the breathing of these premature infants. Now, so we have type 1 and type 2. We also have something called an alveolar macrophage, sometimes referred to as a dust cell. And these are going to be involved with our immune system, our innate, non-specific immune system. These are phagocytes that will phagocytize and bring in different types of debris within the lungs, if there was dust, if there was bacteria or other pathogens. These alveolar macrophages will consume and phagocytize this debris to prevent it from causing damage within our lungs. And then lastly, we have our pulmonary capillary that will be associated with our type 1 pneumocytes to function in the exchange of gases. So here we can see an entire alveolar sac, or part of it at least. You can see your individual alveoli. And then this region on the right, this is just one of these regions expanded upon. So you can see these cells are very, very thin. So these are going to be type 1. And then your type 2, you kind of have to use your imagination a little bit. It doesn't look perfectly cuboidal, but it's roughly cube-shaped, and it's much less flat or squamous shaped than your type 1 over here. So type 1, again, involved in gas exchange with the capillaries, and type 2 will be secreting surfactant that lines this alveolus. Now, surrounding our lungs on the outside, we have our pleura, okay? So you have two lungs. You have a left lung and a right lung. Left lung will contain 2 lobes. Right lung will contain 3, and these lobes are divided by different fissures. So an oblique fissure in both lung, and then the right lung, since there's an additional lobe, you have an additional fissure, and we call that the horizontal fissure. It separates the superior and the middle lobes of the right lung. Again, you have 10 bronchopulmonary segments in each, so roughly 20, and then you have pleura that surround these lungs. And now pleura is going to be analogous to the pericardium in the heart, as well as the peritoneum in our abdominal cavity. And what it is, it's a double-layered membrane. That one layer, the visceral layer, is on the surface of the lung, and the other, the parietal layer, is on the surface of the thoracic cavity. And they serve to surround the lung, and they create a little space, and it's really a potential space that normally doesn't contain a whole lot of anything. There's a little bit of a few milliliters of serous fluid, or what you can call pleural fluid there, and that helps to reduce the surface tension. Again, when the lungs expand, it decreases friction. But there can be conditions in which additional fluid or air will develop within that potential space, that pleural space. So as air is coming down our trachea, it will go down our trachea and branch into our 2 mainstem bronchi. And the point where those bronchi enter the lung is going to be called the hilum. Okay, so the hilum is not specific to the lung. We have a hilum of the lung. We have a hilum of lymph nodes, hilum of the kidney. It's just the general region where structures are entering and exiting. Okay, and then pleura, we talked about these. I'll show a picture in the next slide that shows you exactly what these are. So it must be in the next slide, actually. So the heart is going to be located in the mediastinum, and on either side of the heart, you have your lung. So here you'd have, if this was an anterior view, here you have your left lung and your right lung, and then you can see that on this side. So you can see a right lung with 3 lobes, and then on the left lung you see 1 lobe here, and then you can see part of the inferior lobe on this side. Here's the hilum, where things are entering and exiting the lung. You can see your fissures as well, so oblique, and then since this is the right lung, you also have a horizontal fissure dividing your superior lobe from your middle lobe. Here on your left lung only, you have something called a cardiac notch, and this is specific to the left lung due to the presence of the heart kind of projecting off to the left. That is what this cardiac notch is essentially for. Now, you don't have to know all of these bronchopulmonary segments, but what I do want you to understand is that I want you to understand the association between a division of a bronchi and what region of the lung it supplies. So each primary bronchus, so the right and the left, will get a lung. Okay, so the left gets the left lung, the right gets the right lung. Your secondary, which are also called your lobar bronchi, get a lobe of the lung. So there are 2 on the left that get the superior and inferior lobes, and there are 3 on the right that get the superior, middle, and inferior lobes. You then have your tertiary bronchi, which are also called your segmental. Each one of those gets a bronchopulmonary segment, and that's what all of these colors are representing. So for each lung, you have roughly 10. And now you don't have to know all 10, but just do understand that a tertiary or segmental bronchio will provide air to one specific bronchopulmonary segment. These segments are also important because they can be targeted clinically for a lung resection. So for lung cancer or something like that, you can take out a specific bronchopulmonary segment. So again, you can see your lung hilum here where things are entering, and then in this view in situ, you can see where things are entering and exiting. And then here now we can finally see our pleura. So on this left image, you can see a green layer and you can see a red layer. The red layer, since it's on the surface of the lung, this will be what we'll call our visceral pleura, and then the green layer will be our parietal pleura. But at certain locations, so right here and right here, what do you see? Hopefully you see that the red and the green layers are really continuous with each other. They're not separate. They reflect onto each other. So we call these areas lines of reflection, where you have one layer. So in this instance, we'll have our parietal here in green, and then it will slowly transition into our red, and now we're visceral. But it's really all one layer. And between the two, that's where you have your pleural space. Okay, so you'd have a little bit of fluid. You can see the same thing on this right image. You can see a red layer, which is now going to be your parietal on this side. You come up and it will reflect right here into a visceral, which is blue, and that visceral is on the surface of the lung. And you can see that blown up in this small little image here. Now, we'll stop there for part 2, and we'll pick up with part 3 discussing pulmonary physiology.